Good morning, everyone. Ed Schilling, BMES Executive Director, kicking off the last day of the annual meeting. I hope it's been productive for you. I hope you enjoyed the BMES Bash at the aquarium last night. It was a lot of fun. Really enjoyed it. So to start us off this morning, it's my distinct pleasure to, for the first time, introduce our new president, Dr. Gilda Barabino, for the first time on stage live.
Good morning, everyone, and welcome to our final day of the conference and our exciting plenary session. I'm really excited that my first official duty as president is to introduce this morning's plenary speakers. And we're going to start with the Rita Schaeffer Young Investigator Award. The Rita Schaeffer Young Investigator Award was established by BMES in 2000 to honor Rita Schaeffer, former BMES Executive Director. Rita's gift of her estate, along with contributions for her family, friends, and associates, has enabled this award to recognize the achievements of an early career investigator. This year's awardee, Christian Metallo, is an assistant professor of bioengineering at the University of California, San Diego. He's also a member of the Institute of Engineering and Medicine and Moore's Cancer Center at UCSD. He received his bachelor's in chemical engineering from the University of Pennsylvania in 2000 and conducted bioprocess engineering research at Merck Research Laboratories from 2000 to 2003. Christian received his PhD from the University of Wisconsin-Madison in chemical engineering in 2008, where he studied stem cell biology and tissue engineering. Subsequently, he was a postdoctoral fellow in the laboratory of Greg Stephanopoulos at MIT. From 2008 to 2011, he was awarded postdoctoral fellowships from the National Institutes of Health and the American Cancer Society to apply systems biology approaches to study metabolism and diabetes in cancer. His work at MIT involved the study of central carbon metabolism in tumor cells and its regulation by hypoxia and oncogenic signaling pathways. At UCSD, he continues to apply experimental systems biology approaches to understand the role of metabolism in cancer and stem cell function. On behalf of the Biomedical Engineering Society, it's my distinct honor to present the 2012 BMES Rita Schaeffer Young Investigator Award to Christian Metallo. Christian, welcome.
Thank you, Gilda. When I heard the news of this award and shared the news with some of my colleagues, they uniformly told me how wonderful a person Rita Schaeffer was and how much he meant to BMES and bioengineering in general. So it's an honor to have this award, which bears her name. So if I can start this up. I'm excited to share with you some of my work from the past several years involving the study of metabolism in cancer and other diseases. Before we dive into cancer-specific metabolism, I want to talk a little bit more broadly about metabolism in the context of disease and bioengineering. And one point I'd love you to take away from this lecture is how valuable it can be to understand the metabolic phenotype of the systems we're studying. So we have various lineages and cell types in our body, many of which you all study, and they all are tasked with performing very specific functions. Red blood cells must carry oxygen around, the liver must deal with a lot of all the food and drugs we intake. As a function of these needs, they have very specific metabolic requirements. Furthermore, throughout the course of life, our cells will experience various stresses and different microenvironments and nutrient availability. Now, all of these factors work together to generate a diverse set of functional metabolic phenotypes. Here, depicted here as an illustration of central metabolism behaving differently in different cell types and under different conditions. So it's very important as we try to gauge the phenotype of a disease to understand the metabolic aspects of that phenotype. Now, in terms of cancer, we've known for almost 100 years that tumor cells and really all proliferating cells exhibit very different metabolism than surrounding tissues. So Otto Warburg showed that proliferating tissues undergo aerobic glycolysis. So they take up large amounts of glucose in the body, and nowadays we use that as a diagnostic tool. If you can infuse a patient with a glucose analog, fluorinated deoxyglucose, and use PET scanning to observe the localization of tumors or metastases. But a point that's relevant for this talk is that it's important to understand metabolism in the context of its own microenvironment. And cells in the body have numerous substrates available to them, and they can have different ways of metabolizing those substrates. So I'm sorry to do this so early in the morning on a Saturday, but for the purposes of this talk, it's useful to give a brief overview of central metabolism. So glucose is taken up by cells and phosphorylated. It's metabolized in glycolysis to yield two pyruvate molecules. What Warburg showed is that cancer cells and proliferating tissues will divert much of this glucose to lactate. And we think this occurs to enhance the cell's ability to synthesize intermediates for nucleotide synthesis and amino acids and other cofactors. Now, pyruvate can also be transported into the mitochondria and oxidatively metabolized in the Krebs cycle or TCA cycle. And this is a more efficient way of generating energy in the form of ATP as it's coupled to cellular respiration. Another important point, other substrates can fuel TCA metabolism, and many of these reactions are also catalyzed in the cytosol as well for biosynthetic purposes. So over the last few decades, as we've gained traction in the molecular biology of cancer, we've started to learn that many oncogenes and tumor suppressors, key drivers of tumorigenesis, intimately control metabolic processes. Some of those are listed here. We've also learned that the microenvironment itself exhibits profound influences on metabolic function. We'll hear about oxygen today, but contact with extracellular matrix will also influence metabolic activity in cells. And finally, also very exciting, we've started to learn that many metabolic enzymes themselves contribute to tumorigenesis. Loss of function and gain of function mutations, for example, in TCA cycle enzymes cause cancer. So what we're hoping to do is now exploit the metabolic phenotype of some of these specific cancers and tumors in general to identify molecular targets to mitigate tumor growth. But one necessity for that to come to fruition is we need to quantitatively understand these processes and quantitatively study them. So I'm a flux person, and we like to make an analogy to traffic and flow of cars, which is very analogous. And depicted here is my commute home, which is quite nice with the Pacific Ocean right there. But nowadays, on all of our phones, we can access Google Maps, and we have access to this traffic information. And with the click of a button, we can see how traffic is flowing, what the flux is of cars. And we can see, in this case, if the 5 is backed up, we can engineer a different path, and perhaps a more efficient or scenic route can be used. And this information is very useful as we try to find the best, most effective route home. Metabolism is no different. It's a little bit more complex, and there's some chemistry involved, but this flux and quantitative information is necessary to understand how metabolism is functioning. So the challenge for us as bioengineers is to turn these wonderful maps, which have been painstakingly put together with a century of work by biochemists and turn them into quantitative information on metabolic fluxes, which is the ultimate metric of enzyme function. So we have great analytical tools in the form of NMR and mass spectrometry now to quantify metabolites and, as we'll see, metabolic labeling. But the metabolite abundances can only go so far. If we think of a very simple network here where we see metabolite C having increased abundance, that doesn't tell us specifically which enzyme is dysfunctional, enzyme X or enzyme Y. If we want to develop a drug that targets one of those enzymes, we need to know this information. So to go a little bit deeper, we do a couple things. We incorporate the use of isotope tracers, metabolic tracers, which are incorporated and metabolized by cells into various metabolites, and we analyze this data as a system. So mapping the metabolic network to a stoichiometric matrix with a flux vector subject to steady state and mass conservation constraints. So I'm not going to go into the details of the modeling in this case, and I'll mostly show you some simple model outputs and raw data when applicable. But I'd like to focus on the biological question at hand. Is, in the simplest terms, is how do nutrients contribute to lipid and fatty acid synthesis in cells? And the important metabolite to think about here is acetyl coenzyme A. If you open up your biochemistry textbook, it tells you that carbohydrates are used to synthesize lipids. Glucose is lysed to 2 pyruvate molecules, and this pyruvate passes through the citrate shuttle to generate lipogenic acetyl-CoA in the cytosol. I alluded to before, but glutamine in proliferating cells can also be a significant contributor to carbon in the TCA cycle. Enters at the point of alpha-ketoglutarate, but it can be metabolized in 2 different pathways. It can be oxidatively metabolized in glutaminolysis, which then passes through the citrate shuttle to generate acetyl-CoA. Alternatively, and we've known that this activity exists in cells, but alpha-ketoglutarate can be reductively metabolized in the reverse, quote-unquote, reverse direction of the Krebs cycle in a carboxylation reaction catalyzed by isocitrate dehydrogenases. So one of the overriding questions is how do these different pathways and reductive metabolism in particular contribute to cell growth? In particular, in the tumor microenvironment, where prevascularized tumors, solid tumors, may experience transport limitations in oxygen and hypoxic regions where experience oxygen in the range of 1 to 3%. So the basic way we run these experiments is by culturing cells in the presence of an isotope-labeled tracer, such as glucose. In these maps, these circles represent carbon atoms. The filled circles are C13 isotope labeled, and the open circles are C12. So we culture cells for several days such that this glucose contributes to fatty acids, which enter the biomass pools. We can extract that biomass and analyze it via mass spectrometry, which tells us how many isotopes are present on each molecule and the relative abundance of each isotopomer. And this tells us some very specific information about how our substrate is used. Generally, we can tell how much of the lipids have been newly synthesized, but more importantly, we can determine from the x-axis how much our tracer contributed to the lipogenic acetyl-CoA pool. In this case, we see the textbooks are right. It's about 80% of the acetyl-CoA in cells comes from glucose. Now, we have a really good tracer that can be used to look at this reductive pathway. Glutamine, labeled on the 5-carbon, will enter cells, it will enter the TCA cycle. During oxidative metabolism, I'm not going to walk through all the transitions, but this carbon is lost as CO2. But along the reductive pathway, this carbon atom, this labeled carbon atom, is retained and converted to acetyl-CoA. And we can see in cells growing under normal conditions, we see a minor contribution, maybe 10 to 15% of the acetyl-CoA comes from glutamine, not glucose, through this reductive pathway. If we place cells under hypoxia, though, a profound change occurs, and we see the majority of the acetyl-CoA and lipid carbon is coming from glutamine, metabolized in the reverse direction of the traditional TCA cycle. And these are oxygen concentrations where the cells are still respiring. But we see that the substrates which the cells use to synthesize lipids changes. If we flip this experiment upside down, you saw the normoxic experiment before of glucose labeled with glutamine unlabeled. Under normoxia, the cells use glucose. Under hypoxia, glucose minimally contributes to lipid biosynthesis. But we know these cells grew, and we know they synthesized lipids from the previous experiment. So the model output of this data shows this in more simple terms. We see the percent contributions of glucose oxidation and glutamine reduction under normoxia and hypoxia, and it's really a mirror image of itself. All the data I've shown you so far is relative contributions, but if we measure the absolute flux, we see that glucose oxidation decreases markedly, but we do see an absolute increase in reductive glutamine metabolism. So the cells are activating this pathway. One offshoot of these results is that hypoxic tumor cells may be more dependent upon glutamine than those in oxygenated tumors. And most cells can't grow in the absence of glutamine, but some hepatoma cells can, and we found one of those. In the presence of glutamine, reductive flux increases. And in the presence of glutamine, the cells don't really care if they're under normoxia or hypoxia. Normoxic cells grow more poorly in the absence of glutamine, but this is significantly, their growth is significantly compromised in the absence of glutamine. So it indicates that targeting glutamine metabolism may be an avenue for the mitigation of solid tumor growth. And in fact, some drugs that are currently used, such as asparaginase, which is used in pediatric leukemias, breaks down asparagine to aspartate, but it also exhibits glutaminase activity. So some of these are already being used in the clinic inadvertently. Now, having said that, virtually every cell we've tested behaves in this manner. 4 are shown here from different tissues of origin, including non-transformed lung fibroblasts. So this seems to be a common occurrence in mammalian cells. Even activated T cells, they typically undergo activation in more hypoxic microenvironments. If we activate T cells with a ligand specific for their T cell receptor, do it under normoxia or hypoxia, we see that this mode of metabolism is activated in these situations. So we next wanted to ask what are some of the molecular drivers of this metabolic response. So one of the key sensors of oxygen in mammalian tissues are these prolyl hydroxylase enzymes. They hydroxylate proline residues on hypoxia-inducible factors, or the alpha subunits of hypoxia-inducible factors. So under normoxic conditions, they hydroxylate residues on HIFs, and this von Hippel-Lindau factor is an E3 ubiquitin ligase, ubiquitinates and marks these proteins for degradation. So under normoxic conditions, these proteins are constitutively expressed and constantly degraded. Under hypoxia, by the activity of a reactive oxygen species, this reaction is inhibited and the prolines are not hydroxylated and HIFs are not recognized by VHL. So they stabilize, they can bind a transcriptional coactivator, a nuclear translocator to induce transcription of numerous genes. And this has been a well-studied phenomena over the years. We know many of the genes that are transduced, mostly in glycolytic enzymes and lactate dehydrogenase. But another important enzyme that's a target of HIFs is PDK1. This is the pyruvate dehydrogenase kinase. This phosphorylates the pyruvate dehydrogenase complex and inactivates it, thereby shutting down pyruvate oxidation. So we wanted to ask if this mechanism might be driving the switch in part to reductive glutamine metabolism. So we then tested a compound dichloroacetate, which has been used over the years. It's an orphan drug currently being tested in brain cancer, because more recently it has been shown to mitigate tumor growth in animal models. So this is one of the first experiments that I ran as a postdoctoral fellow, and it failed. I was all excited about this compound, DCA, and I wanted to look at the metabolic effects, and I saw no effect whatsoever. So a couple of years later, we found out, yes, the cells switch under hypoxia and shut this down, so maybe hypoxia is the key trigger to test this under. And what we see under hypoxia, under normoxia, DCA has minimal effects, but under hypoxia, we block the cell's ability to switch to reductive glutamine metabolism and force them to oxidize glucose. Now, the cells actually don't grow very well and aren't happy with this situation. They synthesize less lipids overall, but it's exciting in that it's a drug which mitigates this switch, and it's currently showing some efficacy in animal models and some human studies. So we also wanted to ask what other oncogenic events might be driving this switch between glucose and glutamine metabolism. So another candidate was a clear cell renal carcinoma. Approximately 50% of all clear cell renal carcinomas are deficient in functionality of the von Hippel-Lindau factor, either through genetic mutations or hypermethylation. The VHL protein is lost, and as I mentioned before, VHL will control the stability of HIFs. So these cells, these VHL deficient cells, are pseudohypoxic. They always think they're under hypoxia. Remarkably, if we test a panel of these cell lines, those with loss of function mutations in VHL always prefer glutamine reduction and minimally oxidized glucose, whereas renal carcinoma cells with wild-type VHL behave normally and switch under hypoxia. If we take a functional VHL protein, add it back to these cells, they look normal again. Or if we take a VHL deficient cell and knock down HIF2 alpha, we partially rescue its ability to oxidize glucose. Now, a cool part of this model system is that all the animal work has been done. They discovered VHL as a tumor suppressor about 20 years ago, and 10 years ago, you demonstrated the role of HIF2 alpha. But if you add VHL back to cells or knock down HIF2 alpha, they won't form tumors. So it's exciting in that we see now, after all this time, we see a very profound metabolic change in cells and a dependence on glutamine in these cells, which is associated with efficient tumor growth. So a couple other systems that we've tested out. KRAS is commonly mutated in a number of different cancers. If we express a mutant form of KRAS into non-transformed NIH3T3 cells, in this case, I'm using polar metabolite labeling, where we can look at labeling in citrate and other organic acids. 5-carbon glutamine tracer will generate 5-carbon labeled citrate in the reductive pathway, and we see when we overexpress mutant KRAS, we see a relative increase in reductive glutamine metabolism. So it's also associated with KRAS. And now we're exploring other oncogenic perturbations. Loss of function of the LKB1 tumor suppressor is commonly mutated in about 20 to 30% of non-small cell lung cancers. When we add a functional copy of LKB1 back to cells, we see that glucose and glutamine metabolism, TCA, oxidative TCA metabolism is modulated by adding this energy sensor back to cells. And while these changes don't seem as remarkable in the context of metabolism, these are pretty significant switch that we're seeing. So we're starting to see is that the balance of carbon in mitochondrial metabolism and TCA cycle is tightly regulated by cells and modulated by oncogenes, tumor suppressors, and the microenvironment in general. And the hope is that as we gain more knowledge about how the genotype influences the metabolic phenotype of tumor cells, maybe this can be used to design, to inform treatment strategies that target metabolic pathways. So a question I always like to ask at the end of previous talks was why? Why would cells behave in this manner? Does it really make sense? And it starts to become a little clearer when you think about the microenvironment of a tumor in general. I mean, hypoxia in solid tumors is not the most realistic environment. As oxygen becomes limited, glucose and other nutrients will also become limited, and ischemia is likely to ensue. Furthermore, the cells will see elevated levels of CO2, hypercapnia. So it makes sense that a cell might want to start to conserve carbohydrates and use them more efficiently, and divert flux to pathways which carbohydrates are necessary for. For example, ribose synthesis or hexosamine biosynthesis for nucleotides and glycosaminoglycans, and utilize amino acids and macromolecules to fuel mitochondrial metabolism. So the question then that follows is, where is this glutamine coming from? We're starting to gain some insights to answer this question. If you think about it, the way in which we culture cells is not very realistic, and high nutrient-rich DMEM in liquid media is very different from solid tumor microenvironments, which will be within a matrix of glycosaminoglycans and protein. Also have some of their friends around, which may be dying as well and can be eaten. So we wanted to ask, can protein contribute to metabolism in this manner? So in collaboration with the Barsaghi and van der Heyden labs, we're now starting to answer that question and see, can cells consume and catabolize protein, and can this contribute to central metabolism? So we produced some 13C labeled protein in yeast, extracted that, and cultured it in the presence of more physiologically relevant levels of glutamine in KRAS mutant 3T3 cells. And what we see is, even just after 24 hours of culture, we see many amino acids begin to acquire label in both the culture media and intracellularly. Furthermore, some of these amino acids are metabolized by the cells. So serine hydroxymethyltransferase will give rise to these partially labeled serine isotopomers. So this provides evidence that not only are the proteins being consumed and degraded, but metabolized by cells. Furthermore, we see this labeled carbon enter the TCA cycle, and we can see labeling in citrate and other metabolites, which is reminiscent and suggests activity in certain pathways, such as a reductive glutamine metabolism, oxidative glutaminolysis, and oxidation to acetyl-CoA of other amino acids. So this is now telling us, providing another explanation as to why autophagy and catabolic pathways may contribute to tumor growth and survival. We have metabolomic data from human pancreatic tumors as well, which suggests that glutamine is limiting in these tumor cell types. It also has implications from metabolic modeling as macromolecules start to become substrates that are used by cells as well. They're a bit more difficult to account for in such models. So with that, I hope you can see how valuable some of these tools can be to study metabolic function in mammalian cell systems, and how effective it may be to identify molecular targets that exploit these metabolic dependencies in tumor cells. In this case, we've identified a very different metabolic functionality that occurs under arguably more physiologically relevant oxygen levels. We normally grow cells in 21% oxygen. In microenvironments within our bodies, oxygen levels can be in the range of 1% to 8% commonly. So we see such a significant change in the 1% to 3% range. This may be more relevant to in vivo metabolism than previously thought. So thank you all for listening, and I'd like to thank some of my group members who are slowly growing at UCSD who contributed to this work. Much of it was also done while I was a postdoctoral fellow in Greg Stephanopoulos' lab. And a lot of help has been contributed by collaborators from the Boston area, California, and around the world. So thanks again, and I'd be happy to take any questions.
That was absolutely beautiful work.
Thank you.
Thank you for sharing that with us. So if these tumors are consuming the surrounding matrix protein, and there's a surface area to volume problem, I mean, is there enough available resource, and how does that correspond with the increased matrix density and stiffness you sometimes see around tumors?
Yeah, that's an interesting question, and I haven't done the calculations that would contribute to that. But to be fair, the biosynthetic requirements in terms of just raw carbon of cells relative to the density, I think, are quite low. So I think there may be sufficient in the carbon perspective. And I think this mode of metabolism probably only occurs in a particular window where prevascularized tumors, because vascularization is important, and they must strive to drive angiogenesis to supplement that. But this may just hit a particular time and may only be applicable in some tumor types.
Thank you.
Hi, it was a great talk. So,
Thank you.
have you or are you working with anyone to try to take some of this flux data and put it into quantitative models so that you can make more quantitative predictions about consequences for different changes?
Yeah, so I didn't show it here, but we've done some full central carbon metabolism flux modeling, and to try to get at the question of
Oh, you're off air.
of which enzyme is catalyzing this, IDH1 versus IDH2. Normally I have one of these maps, because it can occur in both the cytosol and mitochondria, and we've performed some flux modeling in those contexts to try to get at some of those specific questions. And now we're applying this to reasonably sized models of central carbon metabolism, maintaining an overdetermined system to look at how metabolism changes in tumors with IDH1 versus IDH2 mutations and some other systems. So yes, we are doing that.
Great, thanks.
Very nice talk, thank you. I wanted to ask about reactive oxygen species generation, especially when you have these very oxidative processes with very unusual carbon substrates. I wanted to ask if you could predict which would be most ROS generating.
The source of ROS is a tough one, but people have studied that under hypoxia, and I think complex 3 is an important source of ROS under hypoxic conditions. And the ROS, it's a double-edged sword, because the ROS is known to, let me find a map, is known to oxidize enzymes in glycolysis and kind of back up flux to try to divert flux through reductive, through the pentose phosphate pathway. And so the ROS can actually help induce this mode of metabolism. But then another problem arises, and where does the NADPH come from? Because this is not shown here, but this reductive reaction consumes NADPH. So then that's another question that we're trying to answer. Where is the NADPH coming from in these systems?
Thank you.
Our next plenary is for the Diversity Award. The BMES Diversity Award honors an individual, project, organization, or institution for outstanding contributions to improving gender and racial diversity in biomedical engineering. The award is given for a broad range of activities, including research, education, and service that improves diversity in biomedical engineering in industry and/or academia. This year's winner is Dr. William Monty Reichert. He's a professor of biomedical engineering and chemistry and associate dean for diversity and PhD education at Duke University. He began his post-secondary education at Gustavus Adolphus College in Minnesota, receiving a Bachelor of Arts in Biology and Chemistry, and earned his master's and PhD in macromolecular science and engineering at the University of Michigan. Dr. Reichert completed postdoctoral fellowships at the University of Utah as an NIH National Research Service Award Fellow, a Whitaker Fellow, and an NIH New Investigator Fellow. Since joining biomedical engineering at Duke in 1989, Dr. Reichert has led numerous research and educational efforts, including serving as a director of the Center for Biomolecular and Tissue Engineering, as well as the Duke Grand Challenge Scholars Program. He's a fellow of the American Institute of Medical and Biological Engineering, of BMES, of the International Union of Societies for Biomaterials Science and Engineering, and the American Council on Education. He's also assistant editor for the Journal of Biomedical Materials Research. Dr. Reichert is the recipient of numerous awards, including the Duke Graduate School Dean's Award for Excellence in Mentoring, the Catalyst for Institutional Change from the Quality Education for Minorities Network, and the Society for Biomaterials Clemson Award for Basic Research in Biomaterials. Monty's longstanding personal and professional commitment and contributions to diversifying engineering are evident in the successes he has achieved in recruiting, mentoring, and retaining critical masses of underrepresented minority students and postdocs. These successes have led to a transformative effect on Duke University and serve as a model for others to emulate. Monty truly embodies the spirit of diversity and of the Diversity Award, and he's most deserving of this recognition. On behalf of the Biomedical Engineering Society, it's my distinct honor to present the 2012 BMES Diversity Award to William Monty Reichert.
Okay, now I have to figure out how to get out of this. All right. Okay, now for something a little different. Thank you, Madam President, for this honor, and thank you for the committee. And this has been an interesting journey, and I'm going to take you through a little bit of it, and hopefully I'll impart some useful information. If you look at sort of a timeline of where I've started and where I ended up, I started out in the macro program in 1978. There were 2 female graduate students and 1 minority student who was actually a Venezuelan, Ernesto Rodriguez. I went to the University of Utah. There were 2 female graduate students there. At Michigan and at Utah, there were no women or minority faculty. I went to Duke in 1989. There were 2 female graduate students and 1 minority graduate student. No women or minority faculty. And now it's 2012, and the graduate program is 40% women, 10% minority. One of those students won the BMES Award, Amy. There's 4 women faculty members and 2 minority faculty members, 2 of which are on the BMES board, and 1 is the department chair. So there's been a pretty organic shift over the years, and I made a few contributions to this, but mostly it's been an interesting evolution. So what I'm going to do now is I'm going to sort of take you through some of my thinking, and I wrote about 5 of these presentations, and so since I couldn't figure out which one to do, I decided to have a presentation with 5 parts. So the first one is thank yous, and there's a lot of people that really went to bat for me when I started this, and but first of all, I have to recognize sort of the irony of getting this award being in Atlanta, and so Dr. King was a huge icon in all of this. In fact, I went, rather than going to the minority luncheon, I went to the Martin Luther King Memorial, and I have my Martin Luther King tie on. But it's really an extraordinary experience, and if you've never done that, I highly recommend that you go. And of course, this is the Ebenezer Baptist Church, which is part of the memorial. I also want to, it's kind of daunting to be the 4th person in line. And one of the things that this award really does is it legitimates or legitimizes diversity as a professional activity, and not so much as a recreational aspiration. And so, so if we look at Sheldon Weinbaum and Cato Laurentian and Madam President, this is a real tough act to follow. And I was trying to think of who these people make me think of. And so it's like following BMES's version of Moses, Jackie Robinson, and Rosa Parks. So, pretty impressive group. So I was thinking, okay, so where am I in this spectrum? Well, I sort of view myself as part Energizer Bunny and part Catalyst. And I'll point these 2 different aspects of my approach out as we move along. I also want to thank the administrators. When I announced after I got tenure that I'm going to do a sabbatical at North Carolina Central University, you could hear the jaws dropping left and right at Duke. But surprisingly, Roger Barr, who was the chairman at the time, he said, sounds like a great idea. He was very supportive. Earl Dow was also very supportive. The provost at the time, John Strobane, was skeptical and suggested that I might consider something else. But then, because I'm there, when it comes to this type of thing, I have no off button. So this is the Energizer Bunny part. So he realized after a while that there's no not doing this. So he became supportive. And then Mort Friedman and George Trusky, of course, were the chairs as I continued to do this. And Lou Siegel and Joey Wright were the deans of the graduate school. And then Jackie Looney is also a dean in the graduate school that were very supportive. So I was lucky that I had people that were willing to let me proceed. I also was able to recruit, as the director of graduate studies, 2 phenomenal students, Laurie Norton and Halawi Betre, who were the first, I refer to them as the prow of the ship, were the first 2 students that really allowed me to pursue this enterprise. And behind there is Jeff Lamack and Jason Smith. And this is in May of 2006, so this is after these students, clearly they got their robes on, so they're graduating. And so I really am thankful for these 2 individuals being hardworking, bright, and persistent. And I was also lucky that I had a number of colleagues who were willing to participate with me. And so here's my Hollywood squares of Duke faculty colleagues who were people that, when you went and talked to them and said, I really think you need to consider recruiting the student, they recruited the student. And so I'm very thankful. So this arrangement of people really allowed this to happen. And I'm also very thankful to the NIGMS postdoctoral training program, which that and funds that I use through the graduate school, I used as a mechanism by which I could bring these students onto campus and get them engaged. And Warren Jones was the director of the program. He just recently stepped down. We have a new director now, and thank God I got it renewed just in the last year, so I'm able to continue this. But this has been a very supportive and very good program. So I have a lot of people to thank for this. Okay. So part two is sort of how are we doing as a community, an education community. And there's a very, you're probably familiar with this, but there's a very interesting document that's out, which is Engineering by the Numbers, and it's put out by the ASEE. And it really goes through in a detailed fashion as to what is the current state of the engineering enterprise in terms of gender and diversity. So in case you didn't know this, I'll show you some data here. If we look at the, if you proceed from the bachelor's degree to the PhD degree, the first thing to notice is that the undergraduate program is by and large a domestic enterprise, and with a very small non-resident component. But then as you move through the master's and into the PhD, there's an overall shrinking of domestic participation that goes on. So you basically are below half when you get to the PhD program. So that's one thing to keep in mind, that the overall composition of the students is diminishing as we go from the bachelor's level to the PhD level. And it's pretty dramatic. The second thing is that if you compare, and a lot of people consider this an inappropriate comparison, comparing the participation of people in the engineering program to their proportionate contribution to the U.S. population, but it's interesting just to use it as kind of a threshold perspective. So what I did is I took this data and then I converted it to a nice little bar chart that's easy to look at. And so if your bars are on the left, that means that you're underrepresented. And if the bars are on the right, that means that you're overrepresented. And so there's a few things to notice here that are interesting. One is that Asian Americans are, we regularly celebrate Asian Americans. We're very lucky to have them. They're a very bright, hardworking, and effective group of scientists. And you can see that they're faring quite well. If you look at African Americans and Hispanic Americans, they're dramatically underrepresented. White Americans are sort of right where they should be in terms of proportionate representation, which I find interesting. And women are fairly substantially underrepresented. So, and this is probably not news to you, but minority cohorts and women do not track in proportion to the general population in engineering education. However, if you look at the, if you do shift the normalization from the general population to the number of students that get a bachelor's degree, you see that that whether or not you're over or underrepresented remains the same, except for females. But generally, it's not very much. So the fractions of students that come in as bachelors tend to progress throughout the program, okay, so even though students leave as we go, the proportionate representation is about the same. So from that point of view, it's interesting to note that it's fairly consistent. So just to draw a few conclusions here, females, African Americans, Hispanic Americans, and other, which are primarily Pacific Islanders and Native Americans, are all dramatically underrepresented relative to the general population in engineering education. White Americans are pretty much where we expect them to be in terms of the general population. Asian Americans are dramatically overrepresented relative to the general population. And the point that I think is more interesting is that the relative proportions of each cohort that receive a bachelor's, a master's, and a PhD degree are roughly the same. So that disparity between the general population and if we look at the group of people that choose engineering is quite different. Okay, well, how about the professorate? How is that doing? And again, this, using this approach, Asian Americans are again overrepresented, African Americans, Hispanic Americans, and females are underrepresented, but it's not as dramatic as, again, compared to the general population. And so if you look at where students are, or I mean, sorry, the faculty are, in the upper left-hand corner are the programs with the largest faculty, engineering faculty. And on the upper right are the Hispanic tenure track faculty and the schools that are there. And then the lower left are women, and the lower right are African American. And I highlighted a few of these with different colored arrows. The green arrows are the schools that appear in all three of the other, in the Hispanic, the African American, and the women's group. So Georgia Tech, MIT, and Penn State are all in each one of these. So kudos to those three institutions. The institutions that have the red arrows for women, Hispanics, and African Americans are all from the group of the largest 10 schools. And the ones that have the blue arrows are minority-serving institutions. So it's interesting to note that minority-serving institutions and the largest institutions are the ones that are most participatory in terms of bringing women and minorities into the engineering professorate. If we look at how BME is doing in terms of women, it's one of the top choices for women. It's up there with environmental engineering and engineering management in terms of faculty options. If we look at African Americans, it's actually fairly far down the list. Petroleum and biological and agricultural are the top choices, and the little blue arrow shows where biomedical engineering is for African Americans. For Hispanic Americans, it occupies roughly the same location, except the top choices are different. It's civil and environmental and engineering management are the top two choices. So to go through the faculty conclusions, female and African American and Hispanic American professorates are underrepresented relative to their proportion of PhD degrees, but not as dramatically as for students. Asian American professorate is overrepresented relative to their proportion of PhD, but not as dramatically again for students. The largest engineering schools, which is a relatively new phenomenon, and the minority serving institutions, not a new phenomenon, hire the most women and minority faculty. And BME is preferentially selected by women faculty, but not by African Americans and Hispanic American faculty, which is an interesting observation. Okay, part 3 is I'm going to get in my soapbox a little bit, and I figure if I can't do it now, I'll never be able to do it. But the main thing here is that diversity is not for sissies. It's not something that you can do and people will be thrilled. So I have a few things I'm referring to as soapy thoughts, since I'm on my soapbox. First of all is that diversity is not part of higher education's DNA. It never was, and it may be someday, but the roots of what we do is a homogeneous and exclusive group of people learning and perpetuating homogeneous and exclusive thought. That's where we got our start. So if diversity in higher education is post-transcriptional modification of their, really the only way that I'm aware of that you can change the conversation in the room is change the people in the room. And you either physically remove them or you change their attitude. And that's the transcriptional modification that has occurred over the years. And so now, and one of the interesting aspects of this and I thought about mentioning this, and Tony Giuseppe Ely said, you should say this, is that I now hear women say things that 20 years ago people used to say about women in terms of, you know, their suitability, people's suitability for certain circumstances. So I'm not sure if that's progress, but it's a change. Okay, soapy thought number 2. Diversity is not really about improving the product outcome. And this is something that we've been hearing in the diversity field for a long time. And it's a really tough sell in engineering. Rather, it's about broadening the group of people who have a stake in the enterprise. Okay, and the reason why this is important is that the sands are shifting dramatically in terms of what the United States is going to look like. And so the orange part is the white Americans, the blue part are the Hispanic Americans, the gray part of the African Americans, the yellow is Asian, and so on. So you can see that the group of people that we're going to draw our talent from is substantially different in 2050 than it is in 2010. And so I think we need to continue to seek opportunities to attract people into the enterprise, and I think that's the main real justification for diversity. Okay, soapy thought number 3. This is something that's perplexed me for a number of years, and this is what, you know, one of the nice things about being a white male diversity officer, which is kind of odd, is that there's kind of a camouflage effect. And so I hear this fairly regularly. I support diversity, but I cannot afford the risk of recruiting in minority students. Plus, the minority problem is never going to get fixed until the problems in K through 12 get fixed first. Okay, so I hear this fairly frequently. And so the sort of the normal views that people have about this is minority education is an onion skin, and you peel off one layer, and then there's another problem. So you look at the grad school problem, the problem is at the undergraduate level, you know, well, undergrads, we can't get students out of high school. High school's annoyed because people get dinged out of the system middle school, and on and on and on. And then you go down to things like nutrition and home environment and parental expectations, and there's sort of like no end to this thing. And so it seems like an unfixable problem. And the other one is this idea that education is a zero-sum game, and there's a lot of stuff that's going on, you know, the Michigan decision and this thing that's going on at the University of Texas and so on. So this idea that there's finite resources, and so faculty may take sort of a risk-averse profile, and even the well-intentioned people among us sort of hedge their bets on recruiting the best prepared students, and so this puts less well-prepared minority students at a serious disadvantage. And so that's sort of common explanation number 2. And the problem with this is it never really fit with, I know these people, and you know, I talk to them and I work with them, and they're my friends, and I just thought, there's something not right about this. And then I had the opportunity to learn about this man, Dan Ariely, very interesting guy. He's Israeli, very outspoken. And his thesis, I condensed it a little bit, but people are mostly honest brokers and who may commit dishonest acts. And there's 2 main reasons why they may commit dishonest acts. One is that the risk-to-benefit ratio seems to be acceptable, but the thing that he's contributed to this is really quite interesting, is that committing the dishonest act doesn't impact your image of yourself as an honest person. And so that creates a very interesting sort of paradox, you know. How can you commit dishonest acts and view yourself as an honest person? And so here's a couple of trivial examples, or a couple examples. The first one is the white lie. No, those pants don't make you look fat. Okay. Okay, so maybe the person does look fat in the pants, but the benefits to saying this outweigh the negative aspects, and it's just a little white lie, so it really doesn't, you're not really being dishonest. And there's more serious examples of this. Committing fraud, cheating on your taxes because I'm already paying too much. Okay, so, you know, chances of me getting caught are relatively minimal, and I'm already paying too much, and the government's a bunch of crooks anyway, so I'm just going to pay less taxes. So you still think of yourself as an honest person, and you're just being, that's actually, in his parlance, that's called being depleted. Okay, so my diversity analog to this is that faculty are mostly open-minded optimizers who frequently decline to diversify their research groups because, one, the risk to benefit ratio is deemed unacceptable, and two, declining the opportunity does not negatively impact their sense of open-mindedness. And I think this is one of the issues that has made it difficult for people to get engaged in the diversity enterprise, even though they're open-minded and well-intended. And so if you go back to what I've been hearing over the years, it's a poor risk to benefit ratio, and the problem is bigger than me anyway, so it allows me to maintain my open-minded self-image for this to remain intact, because, you know, it's just not worth it, and it's, you know, and I'm still support diversity. So this led me to my wacky abstract, and I always viewed the Serenity prayer as a call to action, and you know, God grant me the serenity to accept the things that I cannot change, the courage to change the things I can, and the wisdom to know the difference. So this is an opportunity to do something and make a difference. Where I think what's happened over the years is that Serenity prayer has morphed into this optimization prayer. So God grant me the serenity to accept the things that work in my favor, the indifference to resist the things that do not, and the wisdom to know the difference. And this is a defensive reaction to this. And I think the bottom portion is sort of where we are, and one of the largest barriers to moving forward, even though there has been a lot of progress, particularly for women. So when I first got to Duke, I would say that Duke was in sort of an opposition profile. So there's a direct resistance to difference. Now, over the years, we've moved into what I refer to as the accommodation profile, which is sort of minimum acceptance of difference. People can be different as long as they're not different in the important ways of not being different. And I think where we want to go is more of an incorporation profile, where there's more of a full embracing of difference. And so how do you get there, or what are some of the characteristics? As I said, opposition is where we were in the 1990s. Accommodation is where we are now, and I think we're going to stay stuck here as long as we are a community of optimizers. And we will get to the incorporation profile when affording opportunity takes a priority position. And so that's why my point that optimization trumping opportunity is going to keep us stuck in this accommodation phase. All right, part 4. This is what I tried to do over the years. I tried to lower the faculty activation energy barrier to recruiting minority students. I tried to recruit the best minority students through the normal channels to diminish the distinction. And this is actually important. Students should feel like students. And provides minority students with opportunities to hit the reset button. And this is something that I'm focusing on more now. And so this is the catalyst part of my bipolar personality. And so what I tried to do is match students A with mentors B to produce a relationship A-B by reducing that activation barrier which is associated with the risk-averse thing. And this is by directly talking to people, by offering resources, and by being sort of a co-mentor or someone that the other student can talk to. So that allowed this activation energy barrier to be diminished. And so these are sort of the reaction conditions to making this work. So I think you need to identify a graduate program director who has two characteristics. They have to be among your most productive faculty. You know, the best students are going to want to go into the best programs. And that's true for minority students, it's true for majority students, for women, for men, it doesn't make any difference. And that person also has to recruit or is planning on recruiting minority students themselves. So what you can't see, you can't be. Ask the program director to make direct appeals. Just sort of having a website or a list and identifying students as majority and minority students and just expecting people to say, I'm going to take a student, isn't a very fruitful thing. But if you go into someone's room with a student, this student does what you do, this student is a good student, da-da-da-da, and you sit down, you make a direct appeal to someone to recruit a student, you'd be surprised at the response. Provide resources. This is where the training grant really came in very handy to reduce the perception of risk. Identify minority students are willing to be mentored by mostly majority faculty and then recruit them until things change dramatically. It's majority faculty are going to be the people are going to mentor these students. This is also important. You can't finesse this. This is the not for sissies part. You have to be honest with both parties and say that we'll make it work, but you know, this isn't perfect. And also, if someone is dismissive and just says no, then you just sort of take note and move on. I mean, this is not where you're going to make any progress. So don't try and fix the problem at this level. Use the people that will work with you as your conduit. Okay, so some take-home lessons, and don't try and fix the big problem. You probably can't. Make fruitful student-faculty relationships, one relationship at a time, because that's what we do anyway in our research groups. We make relationships one relationship at a time, so why would minority students be any different? Follow up on a regular basis with everybody, the faculty member, the students. I meet with these students every year. And my own personal bias is it's better to be small and watertight than big and leaky. And I know there's a lot of interest in pushing the pipeline, but one of the worries that we have about the pipeline is, are you able to appropriately accommodate the capacity, or have the capacity to appropriately accommodate the students? And so I think it's really important that if you bring a student in, you make a commitment to the student, and that student finishes and has a productive experience. And that should be the priority. All right, my final section here is some suggested strategies for hitting the reset button. I think this is something that is going to be important in the future because there are a number of students that, minority students in particular, that come into the program. They have a hard time their freshman and sophomore year. By the time they're juniors and seniors, though, they've got it figured out, and they're operating at a pretty high level. Okay, but they've got this baggage of these first 2 years that are going to really prevent them from getting the opportunities that are going to make a difference. And so it places these students at a pretty large disadvantage, even though they've attained the sort of operational level that's suitable. So I think one of the things we have to do, I mean, the summer REU programs have been an important advance, but I think that's actually inadequate experience for really placing students in sort of an optimum profile. And so I'd like to encourage, and now that I'm chair of the diversity committee, this is going to be one of my platform planks here, is encourage faculty members to be receptive to minority undergraduate undergraduates to work in their labs for a couple semesters. And you may have to pluck these kids out of your class because they may not be thinking about doing research. But if you have a kid in your class that asks questions, is clearly on top of the material, go and ask, have you thought about doing undergraduate research? Okay. And in the case of these 8-week things, the faculty member never really develops any ownership for the student, but if they work in your lab in an in-depth experience, then you develop a relationship with the kid. You can write the letter that glows in the dark when they apply for graduate school. So this is really, I think this is really important for students to have this experience. And if we all, faculty members, if you look at who do we recruit in our lab, we recruit students with substantial undergraduate experience. And so this is an important piece of the equation. Another thing that I'm doing is I'm using the master's to bridge the gap between undergraduate and PhD, the thesis masters. And the thesis part is important. Faculty are more likely to admit academic students with blemishes at this point, and students are more likely to give graduate school a try. And what we provide is we provide tuition scholarships to make the experience affordable, and we also provide hourly compensations for students to work in the research lab. And so the students that, had this freshman, sophomore issue, they can effectively erase this experience, have a substantial research experience, and then go forward and apply for PhD programs at a higher level. And right now I have 5 students at Duke who are doing this, and I know some other kids that are at 3 other institutions that I've encouraged to do this. Like another thing we need to do is we need to make mentoring part of the overall tenure rubric. Right now it's the old 3-legged stool. And what I would promote is that we actually make it a 4-legged stool. And we make mentoring part of the assessment. And this would force the faculty to articulate their impact as a mentor. You have to articulate your impact as a committee member. Well, if you can do that, you can do this, okay? And then this is something that's part of the discussion throughout the whole APT process, and I think this would encourage more people to consider mentoring as an important enterprise and would actually have impact. I think Cornell University does this already, and I think it's wise. So my concluding thought is this parable that I actually used in an article that I wrote in 1997 that I think sort of encapsulates my point of view. And so one morning an elderly man was walking on a nearly deserted beach. He came upon a boy surrounded by thousands and thousands of starfish. As eagerly as he could, the youngster was picking them up and throwing them back in the ocean. Puzzled, the old man looked at the young boy and asked, "Little boy, what are you doing?" The youth responded without looking up, "I'm trying to save these starfish, sir." The old man chuckled aloud and queried, "Son, there are thousands of starfish and only one of you. What difference can you make?" Holding a starfish in his hand, the boy turned to the man and gently tossing a starfish into the water said, "It will make a difference to that one." And I think that's really the essence of how you address trying to make a difference, is you make a difference to someone sort of one person at a time. And so when it all works, and this is what it looks like. This is my group last summer, and it's a collection of different but very hardworking, bright, and wonderful people, and you can see they actually look like they're having a good time there. It's been a wonderful experience to work with these people. And I also want to, I had to cobble together quite a few things to make this work, but I've been very fortunate to have access to a lot of resources, and so putting all these things together allowed me to do a few things. So with that, I thank you very much, and if you have any questions, I'd be happy to answer them. So, thank you.
Thank you. My name is Elliot Bodfinick, UC Irvine. I have a suggestion, a quick story, and a question.
All right.
First of all, a four-legged stool is statically indeterminate. My suggestion for minority hiring is to put the young faculty in charge of that. So that's my suggestion. My story is my great aunt, Ruth, she's black, and her grandmother was an emancipated slave. She married my great uncle, who was a Polish Jew, in 1940 in New Orleans. So they ended up moving to Los Angeles. My grandmother was an emancipated slave in Nazi Germany. So in the 40s, there was so much hatred. Now, when you look today at commercials and television shows and around the room, intermarriages, couples of different race, it's just standard. And I think there's a culture amongst my generation that doesn't necessarily see race the same way as perhaps some of the older faculty's generation. So that's my question. Do you see a trend amongst the younger faculty that are more accepting as compared to the older faculty?
This is a really good point, and I think being the diversity assistant dean is a job that eventually I hope will go away because it becomes unnecessary. But I think race matters in that if you look at opportunities available to people, it still seems to track along race and ethnicity. So I think saying that race and ethnicity is sort of morphing into this kind of uniform lump, I think is not reflected in what we still see. Okay. About the youth thing, I started doing all this right after I got tenure. Okay. And now I'm, grizzled veteran status. And if there was a young faculty member in my department who had the same enthusiasm I had, I would hand the whole thing over to them in a heartbeat. But that's not there. So, and I also want to say one thing, is when I ran into Amy at the aquarium last night, who was the student from Duke who won the student award, I said, "Oh, Amy, this is great. Half the students who won awards were women." And she said, "Oh, I didn't notice." I thought, that's beautiful, because in her world, it doesn't make any difference. So things are different. So thank you for that comment. That was a really good comment. Yes, sir.
Monty, along those lines, I was really struck by your sort of pipeline analysis. And it seemed like one of the conclusions that you do support, actually, is that, focusing on STEM in high school and middle school is probably the most effective way to solve the disproportionality in the long term, as just one point. Mm-hmm. But the other one I was interested in was the transition from PhD students to faculty. And I'm wondering whether is that just a time constant problem, or is there really a filtration problem at that boundary? Because, this is a problem with balancing enrollments against faculty sizes. The time constant for students is 4 years, and the time constant for faculty is 40. So is this just, history that's still biting us, or do we really need to focus on the selection process for young faculty?
Okay, let me, I'll take these in order. I'm not anti-pipeline, okay? What I'm saying is that if you want to encourage a faculty member to get engaged in diversity, you don't start out by asking them to fix the pipeline, okay? There are things they can do right where they are that will make a difference, the old starfish routine. And I think asking faculty to approach diversity at that level, recruiting a student, having a student from your class, ask them if they want to work in your lab, these types of things would make a big difference. Okay, but the pipeline piece is, there are things that institutions can do, but I just didn't think in terms of moving the thing forward. In terms of your time constant issue, that's a good question. Everything, the time constant's, 5, 6 years, right? And so that's the data, that's the most recently available data that I had. And so, probably the thing to do would have been to go back 5 years and then look at those numbers relative to the faculty numbers. And I didn't have those numbers available to me. And Rick, you're absolutely right. There's probably some, fudging in there. But yeah, good point. Yes, sir.
Roderick Pettigrew, NIBIB. Couple of comments and then a question. First, congratulations on your award, and it was a very interestingly crafted lecture, very informative.
Thank you.
I want to endorse the concept of a four-legged stool. The more we look at factors that are important in success, the more we appreciate the critical role that mentoring plays. So I absolutely want to underscore the support of mentoring, and I think the educational system as a whole is beginning to appreciate that as well. I also really like the parable of the starfish. The question pertains also a bit of your pipeline analysis, or to your pipeline analysis. I was a little surprised, if I understood you correctly, that the proportion of students that transition from BS to MS and PhD is roughly equivalent for underrepresented minorities as majority persons. That is surprising.
Yeah, I mean, let's see.
And I'll, last statement, and I'll shut up and listen. If you can recall the National Academy's report from about 2 years ago entitled Expanding Underrepresented Minority Participation. As I recall, the conclusion there was that while minorities enter the STEM fields at the same rate as majority persons, the retention of minorities was very disparately low. And so that they advised focusing on retention.
One of the articles that I wrote, this is pretty long ago, the title was Taking Another Look at African American Engineers, the Importance of Undergraduate Retention. And it talks all about this issue, as how a relatively minor change in the amount of retention integrated over all the institutions that are educating African Americans would have a profound effect on the number of people that persist in the program. And I too was surprised, if you look at the proportions of students at the bachelor's level, the proportions of students at the master's level, this is US citizens only, and the proportions at the PhD, it was really surprising that those proportions were relatively consistent from one phase to the next. I mean, they're clearly smaller pieces of the pie, but proportionately speaking, they're relatively consistent. So encouraging more of the bachelor's students to make the step up, I think, could also have pretty substantial. So, I mean, and again, I don't want to be dissing the pipeline. I think the pipeline is important, but I think blaming the whole issue on the pipeline without paying attention to what we can do in our own backyard is inappropriate, in my humble opinion.
Monty, that was a great talk.
Thanks,
We've seen like a big increase in the number of female students, but there's still not that many women in our faculty. Do you have any thoughts on like adjusting the tenure process to increase, work-life balance or make it easier to have children? Or do you have any thoughts on that?
Oh my God. Well, I don't know. I should ask Karen or someone like that to answer this question. My sense is that academics for women is a tough sell. And I think companies, when they recruit people, they do their best to facilitate their job with other aspects of their life in terms of childcare and flex and this and that. And academics, they're, you go out to lunch with the other woman on the faculty and she said, I can do it, you can do it. And that's sort of like your gender program. So I think it's a tough problem, and there's a lot of smart women who are faculty, and I defer to their wisdom. But I think this is something that is very much a work in progress. And so I don't know, Shelley, somebody, one of you guys, maybe you could rescue me. But so I'm sorry, Brad.
That's okay.
But I will say though that the progress has been dramatic in terms of women's participation. Now, encouraging them to go into academics, the more women faculty you have, the more they can, who knows? There might be a coupling there somehow. I don't know. That's a great question. So, lousy answer, great question.
Morning, and thank you, Dr. Ritter. I have a concern.
Okay.
I had this concern ever since the beginning of my PhD program. So, as in this trend, we've seen almost all schools are adding new major, like new sort of field, and also recruiting professors, like more and more professors to fit in these fields. Of course, in turn, these professors will supervise more students, including PhD students. So a trend I have seen is the country as brought to the international, so are culturing more and more PhD students. And what I've seen is we add to certain, competitions, make these competitions more intense in terms of the future career, either in academia or in industry. But, well, still, I think the society is still keeping like the old idea. PhD students or someone have a PhD degree should be a leader in no matter what field. But our PhD students are actually trained in a very specific research field. We are not multiple talent or we're not multiple skills. So I kind of lost my direction. What the so many PhD students we culture to do in the future. Are we supposed to only add to the intensity of this competition, or should we, maybe drop the whole expectation to the society? The PhD student not only to be a leader, but can be a server in the future. So I would like to listen to your comment.
Wow, that's a well, I think the paradigm within a PhD program is to encourage someone to become a true expert in a finite area, and that they use that as the way to learn how to do the whole process. So broadening the experience would come at the cost of depth, and I think that's something we need to be very careful about. In terms of broadening in terms of the professional sense, where's Brad? Brad is very, you should talk to Brad. Brad, raise your hand.
Should we have like,
okay, so we have this thing called the PhD.
Should we control the number of PhD students to save all the sources to culture really elite of those populations? Or should we drop the whole?
Well, what we have is we have this thing called the PhD Plus program. And this is a student-run activity. And what the students do is they get together and they come up with ideas. What are the types of things we want to learn about? What are the types of things that are our concern? They put together workshops. It's a completely student-run activity. And I think encouraging students to take greater ownership for their development and giving them the resources to pursue it is the way to answer this thing. But asking a faculty member to diminish their expectation that you pursue a problem in great detail is going to go nowhere immediately. So I think there are other ways to get what you're looking for and other ways to get information about opportunities, but I think you need to get together with your fellow students and develop something. And I think talking to Brad, he's running the program at Duke, would be more useful than, whoops, again, my lousy answer. So anyway, it's a good question.
My question's brief. And people come to engineering because they know one, or there's someone in their family, just like all the professions, I suppose. They also come because guidance counselors tell young people that if they're good in math and science, they should be an engineer, which is sort of a mythology thing in a lot of ways. To the degree that participation of any group in this profession springs from somebody getting interested enough to pick this direction, what are your thoughts on the image of engineering in society and the idea, the thing that people come to a profession for is respect, and engineers don't get respect in society like other professions do. We don't have a TV show, we don't, or we only have a TV show about nerds, which is like really not very respectful. So your thoughts on that?
Well, I don't know. I mean, I think any profession that has a 2% unemployment rate right now has somebody's respect. But in terms of, there's no ER equivalent in engineering, and so I think that would be the world's shortest pilot show ever. But I don't know, I mean, you're right. I mean, you don't, exposure to engineers, and it's kind of too bad. If you look at sort of the space program and these types of things, that was when being an engineer was, Sputnik and all that kind of stuff, and that sort of has diminished. But maybe, I don't know, maybe that'll come back somehow. I don't know. The space program's tanked right now. But anyway, thank you very much.
So at the conclusion of our session, I'd just like to thank again our speakers. Chris, that was a wonderful talk. It was very insightful, and thanks for sharing your work with us. And Monty, thanks for saying the things that no one says. Often a lot of these things go unspoken, but we need to talk about them, and we thank Monty for helping us with that dialogue. So thanks, everyone, and I hope you have a great rest of the day and a conclusion to the meeting. Thank you very much.